Guys, in this video for Unit 2, we're starting off learning about cell structure and function by covering section 2.1, which is all about subcellular components. It's basically like a brief overview of all the parts of the cell, and then you dive deeper and deeper in later units. So it's a great foundation for later units. And I know I said that about Unit 1, but Unit 2 is more about the parts, and then you go into the parts and understanding the parts of the cell and how they interact with one another in later units as well. So just so you're aware, this is like we said, a brief overview, cell structure and subcellular components, which is basically the coverage of section 2.1, so the first initial section. Please note the disclaimer below before we begin. So let's get started. So you can see here in this video, we have a brief introduction to cell structure. So I think this table really summarizes what we're going to be talking about, not only in this video, but future videos as well. It's a brief overview of the cell parts, so the parts of the cell. Now we're going to be talking about different types of cells, so I want you to be aware that not all of these parts are in all cells, and you will see that later on. But we did have the cell parts and the functionalities, and we're going to go in more detail. Now let's talk about the cell membrane or plasma membrane. So cells usually have a circular-like shape, not always circular, not a perfect circular, but basically I should say enclosed space. Please note that this is not a perfect circle. All cells come in different shapes and sizes, but we like to believe that in general it is an enclosed space. So I want you to think of this picture of a cell that I have drawn out. And I want you to think of the idea that you are inside that circle. This is your room. And you have a sibling that just likes to come in and out of your room. And you get annoyed and you are like, please don't come into my room. So you have a barrier that you placed in between you and that sibling from coming inside that room. You set a boundary. And so what that is your cell membrane. And what that does is it provides a barrier from what is inside the room or inside the cell from what is outside the cell. And it protects everything inside and outside. But it can also be selectively permeable. So what we're talking about essentially is in the cell, we have this circular-like or covering up shape that's on the outside that provides this barrier, and it is essentially called selectively permeable. So that could be like, hey, I don't want a sibling to come in, but I really want my friends to come into my room. That is going to be selectively permeable. You're only allowing certain things in and out of your room, in and out of the cell. So that is what the cell membrane does. It allows only certain things to go in and out, and that is called selectively permeable. And it also provides a barrier by doing this, by allowing only certain things to come in for safety and protection. The way that it does that is if we were to take a look at the cell membrane and we were to zoom in and cut a slice, it would look very similar to the picture right here where we have inside and we have outside of the room. And what happens is on the inside and the outside of that slice, they have the quality of being hydrophilic. And so it really loves, if you remember, it really loves water. It is water friendly over here. So it's going to be water friendly on the inside of the cell and water friendly on the outside of the cell. However, on the inside, deep within, I want to say, that barrier, what's going to happen is it's going to be hydrophobic. Phobic meaning phobia or water fearing. And so it does not like water in there. And what we call that slice, it's called the phospholipid bilayer. And if you remember, in our macromolecules unit, this is where it all comes in. In Unit 1, you see the term phospholipid or phosphate, you might see the beginning, and then the term lipid, I should say. So you see the beginning of phosphate or phospho, and lipid. And lipid, if you remember, is a type of macromolecule that we spoke about. And if you remember, those are things like fats and oils, and they're really water-fearing in there. They separate from water, and that's how you can remember phospholipid bilayer. The lipid essentially is a bilayer preventing the inside and the outside from really clashing with one another. It creates this barrier, and it is water fearing on the inside. So why is this important? Let's say that we have something that's inside the cell that is water loving, this little blob right here, and it wants to go to the outside of the cell. So it goes through the door and it says, I don't really like being in this water fearing place. So it's more likely to go to the other side because it wants to stay away from the hydrophobic arena. So that is what the purpose of the phospholipid bilayer is and why it has those lipids on the inside. Now, I also want to preface this by saying that it is embedded with different items, one of them being proteins that are embedded inside. Now, I know this picture doesn't have those proteins. There are different types of proteins, and there is a whole unit dedicated to this. For now, you don't need to know the details. You just need to know, guess what? There are proteins that are embedded in the phospholipid bilayer. And essentially, in later units, we're going to talk about those proteins. We're going to talk about how they provide transport, signaling, and recognition. You're also going to understand the carbohydrates that are involved in the phospholipid bilayer as well. For now, that is what these structures are. That sort of has the six-ring structure. If you zoom in, you will see that it looks like, something that is similar to a carbohydrate based on that six-ring structure, which you don't need to know details about right now. You just need to know they're there. They're part of the phospholipid bilayer. And then they have cholesterol that's involved as well. That is somewhere in there as well in the structure. I think it's that. I can't zoom in and see, but one of these is the cholesterol, and the reason and the purpose of that is for membrane fluidity. So just know for now, you have carbs, you have cholesterol, you have proteins. They're all there. They're embedded in the phospholipid bilayer, and they do serve a purpose that you're going to follow through with later units. For now, you just need to know it provides a selective barrier and it provides these functionalities mainly as a source in terms of the plasma membrane or cell membrane. Now that we've covered that, let's talk about the powerhouse of the cell. So I have a picture of what is called the nucleus here. And if we were to take and draw a picture of the cell, we have that big circular structure. We have the plasma membrane or the cell membrane on the outside. And in pictures, you'll see this giant structure on the inside that really takes a large chunk of space. And that is considered the DNA, containing the DNA, I should say, and that is called the nucleus. The nucleus is the largest organelle. We call them organelles, the stuff that's inside the cell. And the nucleus is considered to have DNA. And if you remember, in the macromolecule units, the term DNA is deoxyribonucleic acid, which is a type of nucleic acid, which is a type of macromolecule. See how it ties in together there? And so what ends up happening is the nucleus holds that DNA. And in later units, we're going to cover how DNA turns into RNA, which turns eventually into protein. But for now, you just need to know that, hey, guess what? The nucleus houses that DNA, and that is a very large organelle. Because it's considered the large organelle within the cell, we consider it to be the control center. And by that, we mean like think about your home and how it has electricity. And a bunch of homes in your neighborhood can have electricity, but they are turned on by a power grid system that's somewhere in the middle of nowhere. And that would be the control center that exists for that neighborhood. That's the same thing that we think about with the nucleus. The nucleus holds the power of the cell. It dictates the way the nucleus acts or reacts. And so you could think of the nucleus as a brain. There is also inside the nucleus, there is a small part. So we have the nucleus, which is the structure, and then there's even in there, there's a tinier part. So if you look at a cell, if we were to draw it out here, we have that large structure of a nucleus. Then you can see in some pictures, not always, they will show a mini structure inside, which dictates that part of the nucleus's space. And that is called the nucleolus. And you're going to learn later on that's used for ribosomes and why we need ribosomes. But for now, you just need to know the nucleolus is housed in the nucleus, and it has, or I should say, it has the functionality of creating ribosomes. Now let's talk about ribosomes. Ribosomes, as we previously stated, are associated with the nucleolus. So let's talk about the different specific types of ribosomes. So when we talk about ribosomes, you will see here, there's the picture of that cell again, but it's a better picture than the one I've been drawing so far. So let's recap before we begin. So remember, we talked about the cell membrane, which is on the outside. We talked about that nucleus, which is the largest organelle in there. And then inside the nucleus, we talked about the main structure inside that nucleus, which is the nucleolus. Okay, so now we're going to talk about a little bit of what's this structure here, and I'm drawing two arrows because these structures are the same name, so they're called the endoplasmic reticulum. So the endoplasmic reticulum is this entire structure here. So that's the endoplasmic reticulum, the yellow into the purple. It's not always going to be those colors, but I just want you to notice that difference because you'll notice these little dots on here. And those little dots are considered ribosomes, and those are bound ribosomes. So they're attached to the endoplasmic reticulum, which is this structure that they draw out here as well. So I just want to point that out. If you're given a picture of a cell, it's somewhere like around here. And the bound ribosomes on the endoplasmic reticulum are considered to be different than the free ribosomes. And the free ribosomes are free floating in the cell in what's called the cytoplasm, which is the space where all the various organelles fill. So that is called the cytoplasm. So the free, which is not bound to the endoplasmic reticulum, is different. So with the endoplasmic reticulum itself, there are two different names that we're going to go more in detail again later. But the reason why this is called rough endoplasmic reticulum and this is called smooth endoplasmic reticulum is because the smooth one doesn't have any ribosomes attached to it, while the rough one has endoplasmic reticulums or has the ribosomes, I should say, attached to it. So we're going to cover that later on, but just for now, you need to know that ribosomes can be free floating or bound. They're also the site of protein synthesis, which you might be like, okay, what does that mean? So we're going to cover this later. It's called the central dogma. And what happens is the DNA, which is inside the nucleus, eventually will convert to RNA. So the DNA ends up becoming RNA. And the process of DNA to RNA is one process that goes from inside the nucleus to outside, eventually becoming protein. So that process is called translation. So for now, all you need to know is guess what? Ribosomes assist in protein synthesis, and they're made up of what's called rRNA. You can think ribosomal RNA. That's what the R stands for. Great. Now that we've covered ribosomes, let's go back to that endoplasmic reticulum. And you already know this organelle because we sort of covered it a little bit earlier, where we have the smooth ER, which doesn't have the ribosomes bound. And the bound ER causes the rough ER to exist. So that's what makes it rough. Now, with that information, we now know that there is a difference between the rough and the smooth ER. But that also means that they have different functionalities as well. For the smooth ER, it's known for detoxification, calcium ion storage, and lipid synthesis. So it produces lipids, it detoxifies. Basically, if you think of, usually when you see infomercials where they say this juice will detoxify you, guess what? Your cells in your body already do the detoxification. It naturally does that through the endoplasmic reticulum, through the smooth endoplasmic reticulum. And you also have calcium ion storage as well. So that is the smooth ER. Then you have the rough ER. So the rough ER is where proteins are synthesized and transported through. So that's where the previous term ribosomes, and that's associated with protein synthesis, or we talked about that in when we're talking about ribosomes. Well, that would make sense because they're rough and they're attached. The rough ER is caused by the ribosomes being attached to them, which happens to also be associated with protein development. Okay, so we have protein synthesis and transport for rough ER, and we talked about smooth ER and the difference between the two. Let's talk about this structure, which is called the Golgi apparatus. I want you to think of the Golgi apparatus as a chained mailbox store. And you might be like, what is she talking about? If you take your mail, let's say you have a package and you take it to a post office center, the post office will take the package. Either they will close up your package, they will seal it, and then they'll send it to the appropriate person that it needs to go to. That's what the Golgi apparatus is. It is like the post office. So I think of it as the post office where everything needs to go in and out of the Golgi apparatus before either entering or exiting cells as a final check. So that's what the Golgi apparatus does. There are two locations or faces, cis and trans. So cis means it's going to come in through that location and come out trans. So come out on the other side. So when we talk about a cell, I'm going to draw a picture of a cell here. And say we have my wonderful Golgi apparatus here, and say for some reason the nucleus needs to release a protein. The protein isn't just free floating, it is oftentimes seen to be in like a bubble-like structure called a vesicle. And that's what you see here. And it will literally tell the vesicle, hey, me as the nucleus, I want this out of here. But before I do, I have to make sure to send it to the post office, which is your Golgi apparatus. So it'll send it to the Golgi apparatus through the cis side, and that vesicle will come in, and then the post office will say, you know what, I'm going to fix it, I'm going to store it, I'm going to ship it. And then I'm going to release it to be exited out into the trans side to then release outside the cell. So that's the main purpose of the Golgi apparatus. It is like the post office of the cell. Now let's talk about the mitochondria. The mitochondria is notorious in the animal cell, not the only type of cell that has mitochondria, but it is very important because there's a whole unit dedicated to mitochondria. So I just want you to keep that in mind. The mitochondria, just a fun fact for you, it has its own DNA. You don't need to know that, but it has its own DNA. And why that's important, which we'll cover later on, is that DNA actually comes from your mother, not your father. So anytime that you take a deep breath in, please note that that is due to the mitochondria, which it is involved in respiration, which is also in a later unit. But for now, just know as a fun side fact that your mitochondrial DNA comes from your mother, not your father. And the mitochondria is notarized as being the site of ATP production. It is in this whole cellular respiration section that is its own unit. But for now, you just need to know mitochondria. There's a unique structure involved. It is a double membrane. So there's an inner membrane, which is inside here, and then has an outer membrane, which is the outside part here. And with that being said, there's a matrix that forms inside, and these little folds or crevices of the matrix are called the cristae. So the matrix itself is called a matrix, but the little folds are called the cristae. So it has a double membrane structure. It has a cristae and matrix. It has that double membrane structure is inner and outer membrane, and it is notarized as being understood as being associated with respiration, which is involved in ATP production. And if you remember, ATP is another way of saying energy. So anytime they give you a question, if it's a multiple choice question, and they let you know, hey, we wanted something and it was not able to produce energy, I want you to think, aha, so there must have been something wrong with the mitochondria because it couldn't produce ATP. So that's just a tidbit for you if you get a free response question related to ATP. Okay, let's talk about those vesicles that had the little structures inside that we talked about with the Golgi apparatus. Sometimes those are called lysosomes. And the inside of a lysosome is extremely acidic, and that's so that way it could hydrolyze the foods, the amino acids, whatever's inside. It breaks down any material. So remember, we talked about the post office being the Golgi apparatus. Well, if we were going to talk about that being the Golgi apparatus, I would say that the trash center, which tries to figure out what to keep or what to throw out and recycle, is the lysosome. And the lysosome basically recycles and digests and see what it can reuse back in the cell or needs to go. It is also associated with apoptosis. Apoptosis is another way of saying programmed cell death. Fun fact for you, all cells need to die. The reason why is because it allows for new cells to be produced. So each cell has a life shell, and that is called apoptosis. And that allows the cell to rupture and release acidic content. If we don't have apoptosis, you could have other issues, such as this is what leads to, in some cases, cancer. So just be aware that lysosomes are good for us because they act like a recycling slash trash center that decides what particles need to stay and what needs to go. And it's involved in apoptosis. I also want to draw your attention to this other word called phagocytosis. There's terms like phagocytosis or pinocytosis that you may see, and the cell itself actually can sometimes engulf products by forming this like mouth like structure. I'm a little bit older, so I think of it as a Pac-Man. I'm pretty sure not everybody knows what Pac-Man is, but it forms this like mouth-like structure that allows things to go in and out. And when it engulfs in that way, it's called, if it's a large item, it is called phagocytosis. If it engulfs very tiny particles, it's called pinocytosis. So you want to think of pino meaning tiny, and phago means giant. So that's what lysosomes, how they engulf their food products. Okay, let's talk about vacuoles. Vacuoles are a unique structure that is commonly found in plant cells. So I want you to remember and associate vacuoles with plants because more than likely, if you were given a question related to vacuoles, it would be noting that it is associated with plants. Now, with vacuoles, they take up a large chunk of space in the plant cell, and that's because they store a lot of water and ions. Remember, us as human beings who have animal cells, we constantly drink and eat our food and let go of our food and water. That's not the case with plant cells. They need to store water in case there are seasons where there's not water available to survive. And that's what vacuoles do. Now, they have a sometimes what's called a contractile vacuole, which allows for the expelling of water. So be aware of that. They are very large in structure as well. So those are vacuoles. And another plant organelle is called a chloroplast. I want you to think of chloroplast as equivalent or opposite to the mitochondria. So while we as human beings have mitochondria that use the mitochondria to take our food and convert it into energy, Chloroplasts take the energy or the ATP from the sun. So they take it from the sun and they take that energy and convert it into usable food. So while we eat our food, chloroplasts, which are found in plant cells, are going to go ahead and take the sun's energy, the ATP from the sun, and convert it into usable food. And that process is called photosynthesis. So chloroplasts are the site of photosynthesis. They are the opposite of what mitochondria do. So mitochondria, I want you to think animal cells. Photosynthesis, I want you to think plant cells. Now, just like the mitochondria, they have a double membrane structure. So you can see there's this outer structure and there's this inner structure inside. And they're composed of two main parts, which is called the stroma. So the stroma is like a fluid-like space that is between the outside and the internal membrane, which is called a thylakoid. And the thylakoid are these flattened discs. Now we're going to talk more about the thylakoid in later units once again, but for now you just need to know they're just flattened discs and utilized to help assist with basically collecting that light. Now, chloroplasts are typically green, and the reason why they're typically green, or I should say, why leaves are typically green, is because of the way chloroplasts act. They are there for pigments of the pigment of color is there based on the light absorption. So in certain areas of the US country, we could see that there are leaves that change in the fall, and that's because of different chlorophyll pigments that allow for that color change in the leaves. So think of chloroplasts basically collecting that light and utilizing that light in certain ways that they could change the color and essentially use that energy to convert for food. Okay, now we have centrioles. Centrioles are a very small structure. Once again, just like the other parts, we're going to be talking about it in its own role, in its own unit. But for now, you just need to know that it helps cell divide. See, what happens is one cell can divide into two cells, and that is done by assisting with centrioles. They also produce what's called microtubules, which has its own functionality. But for now, if you see a structure like this with these like tube-like structures, think of it as centrioles and note that it is for cell division. Now let's talk about the difference between plant and animal cells. This is a high yield topic. You might get it in a free response question or a multiple choice question, either directly or indirectly. I want you to think of this picture here, this table here, as your holy grail to understand the difference not only between plant cells and animal cells, but prokaryotes. I want to make a notation here because this was conducted before in a previous test question. I want you to note that plant cells and animal cells are called eukaryotes. So I'm going to put a giant E. And they are separate from prokaryotes. They're two separate main characters. So if we were to think of it, we could say that there's all cells and we would call them X. And then they separate into two categories, which are prokaryotes and eukaryotes. And eukaryotes further break down into plant and animal cells. This is important to note because sometimes they can give you a question where they ask you to compare the difference between eukaryotes and prokaryotes. And people get confused because they think that they're thinking about asking a question related to animals versus prokaryotes. And that is not the case. So I just want to point that out as a mini tip or secret that plant cells and animal cells fall under the category of eukaryotes and prokaryotes are their own separate group. So let's jump down here and take a look at the unique properties of prokaryotes. Prokaryotes, we didn't talk about much in this video, and that's because they do not have membrane bound organelles. So they don't have those membrane bound organelles. So they don't have like the nucleus. They don't have the Golgi apparatus. All those organelles we talked about does not exist. So prokaryotes lack those membrane bound organelles. They usually possess some sort of flagella. And so what that means is if you have a cell here, they sometimes have a little tail And that's to help them to move. They're usually moving around based on some sort of like external hair-like structure that allows them to move. Their DNA is very simple, and they produce or reproduce through what's called binary fission. So I want you to note that they're very simple structures, simple type of cells, where plant cells and animal cells are a little bit more complicated because of those specific organelles. Some even believe that plant cells and animal cells actually evolved from prokaryotes. Whether or not that's true is not necessary to know right now, but just keep that in the back of your head when you're trying to compare between plant and animal and prokaryotes, or I should say eukaryotes and prokaryotes. Now that we've finished talking about prokaryotes in general, so these three bullet points, I'm going to go to the first two to distinguish the differences between plant and animal cells, which are going to be eukaryotes. Please note that plant cells tend to be larger in size, so they tend to be very large. And then plant cells have different types of organelles in terms of the fact that plant cells have cell walls. Animal cells don't. And so plant cells tend to be more rigid. And then also plant cells have chloroplasts because they need that to function with converting the sunlight into energy source, so photosynthesis. And they have a large vacuole, which allows them to store their food or their water in that vacuole. And because of these large cell wall like structure, they tend to be cube in size because it's so rigid. The cell wall tends to be like making it more structured in a very, rather than free floating in a circular or weird like shape for animal cells. So just keep that in mind. And the similarities between plant and animal cells, which you are expected to know as well, is that they have these organelles that we talked about. So just as a disclaimer, while plant cells don't utilize their mitochondria for their form of energy conversion, they do have mitochondria. It's just not their primary source of energy use. So just keep that in mind when you're answering any question, because that is a common trick question. And students often think, oh, well, if I think of mitochondria, it's got to be an animal cell. And while that is true, they're also in plant cells as well. They're just not utilized. So I want you to keep that in mind and keep in mind these common structures or organelles that exist in both plant and animal cells. Okay, now it's quiz time. We're going to take into account one multiple choice question, see how we answer it. So I want you to take a look at this question you see here. Take a moment and pause the video and try to see if you think it's one of these four option choices. So it's option choice 1, 2, 3, or 4. Is it in the cytoplasm, on the inner plasma membrane, on the outer plasma membrane, or in the nucleoid region? So take a moment, pause the video, and unpause once you've selected your answer. We'll go through the answer together in just a second. Okay, now that you've completed pausing the video and trying to come up with the answer, let's see what the correct answer was. The correct answer is on the inner plasma membrane. So the question asks, the chemical reaction in photosynthesis are almost the same in eukaryotes and prokaryotic cells. So I want you to think to yourself, hey, they're comparing eukaryotes to prokaryotes. But I also want you to think, based on the information they gave us, they let us know that they're talking about photosynthesis. So while they're comparing eukaryotes and prokaryotes, they are letting us know with a hint that they're actually talking about plant cells. They never specified in the question that they're talking about plant cells, but this is called deducing your answer. Like you're supposed to deduce the question and read through it to try to come up with a reasonable answer. And in this case, you're told that there's photosynthesis. It's a trick, or it is a way to help you out in a way. Either way you think of it, they're letting you know that there's photosynthesis, and they're letting you know, hey, I know that you understand there's eukaryotes versus prokaryotes, but remember, eukaryotes, there's two subcategories. categories. And in this case, even though they're talking about a eukaryotic cell, you have to deduce that they're specifically talking about plants, not animal cells, because they're mentioning photosynthesis, which we now know takes place in the chloroplasts. So we also know that they mention this again because they're saying something about light-dependent reactions. Please note that we will cover light-dependent versus light-independent reactions in a later unit. And for this specific question, it might have seemed out of context, and that's okay because you don't understand what that means. That's fine. This is a way to help you understand that you could be given questions, specifically multiple choice questions or free response questions, that can have information on there that you might have never seen before. And you have to come up with a conclusion based on the information that's provided. So in this case, they let you know that there are reactions that occur, but they need light. And remember, humans, which have animal cells, don't need to utilize the light to convert to energy. And they also mentioned the thylakoid membrane and the chloroplast. So they're hinting at you that, hey, you know what? We're talking about plant cells. So they're saying, guess what? There's photosynthesis in both eukaryotes and prokaryotes. But I want to know where is going to be the reaction if this is photosynthesis that occurs in prokaryotes, in this specific prokaryote. So it's sort of a scientific reasoning question where they say there happens to be a prokaryote, and guess what? It happens to be very similar to a plant cell, and it happens to have a chloroplast. Well, we know that there are thylakoids and there are these disc-like shapes, and those disc-like shapes are there to catch the sunlight. And so it's going to be on the inside, not the outside, because of the thylakoid that we mentioned very briefly. And it is going to be in the inner plasma membrane based on the fact that they mentioned the thylakoid. Now, I understand that prokaryotes don't have membrane-bound organelles, and this can be defeating for this question. So just remember that they're trying to mention some sort of prokaryote that has something very similar to that. So they're going ahead and saying, we have a very similar structure, and we want to know what that would mean for the prokaryotes. It doesn't mean that the prokaryotes have a chloroplast. And I just want to point that out. They're not saying to you, hey, guess what? We have prokaryotes and they have chloroplasts, because we know based on the information that we have learned that prokaryotes don't have membrane-bound organelles. However, they're letting us know there's something in there that provides us the opportunity to act very similar. So let's compare the chloroplast to the prokaryote. Guess what? We're comparing the inside, which takes the light and converts it, which is the inner plasma membrane, versus the outer plasma membrane, and converting that light. It's not going to be in the cytoplasm because that's not a similar structure. It's not going to be in the nucleus, which is in the nucleus. It's not going to be in the outer membrane because that has nothing to do with conversion of light. And just to go back to the nucleus is similar to that of a nucleus. And the nucleus just basically is the brains of the cell. It's not going to go ahead and specifically have light-dependent reactions. So the best fit answer for this question is in the inner plasma membrane. Now, I know some might be defeated in this question because this question might be allowing you to think, well, I chose a different answer, but this one seems better. And that's the tricky part of AP biology or college biology. You're not expected to choose one out of the two, which might have happened when you were reviewing this problem. The better choice based on the information provided is the better option. And I hope this helps in your review. I hope to see you in the future as you subscribe to this video, and we hope to see you back in preparing for AP Biology or College Biology. Good luck, and we hope to see you soon. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, A homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score by hitting the subscribe button. The more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams. We can't wait to help you succeed.